The topic of my talk today is network construction and inference as an elegant approach to understand disease pathogenesis. Right. To begin with, first of all, I wanted to define or explain what do we understand by systems biology, because till you understand systems biology, this is the subject. This is a broader umbrella under which we study about networks. As Mr. Barua has given you an introduction about data and the data from biological sciences, and how those data can be used for getting meaningful insights from those data and do certain predictions, etc. So that is one aspect of data science in or big data in biology today, where we use AI ML techniques. And the other aspect, other end, is where other application is rather where we use systems biology, modeling, and simulations, or network analysis, or network studies to get inferences. So the objective is more or less same in both the cases. Where in the previous one we get insights and do predictions using AI/ML methods. Here we use various mathematical and theoretical modeling strategies, or network or topological analysis for in systems biology to get those insights, right? So there are two ends, there are two ways by which you can dig out meaning from those big data in biology. So that you, as you all know, there is a huge data explosion everywhere in biological sciences or biomedical sciences. And those data are generated by using high throughput techniques, various high throughput techniques, like for example NGS, then mass spectrometry, etc. Then these data needs to get some meaning, right? So otherwise we if we don't get any conclusion or inference from those data, analysis is of no use. So that's our main aim. So systems biology is one such area. Though it is not a very new field of science, it has been studied, it has been studying in this has been you researchers are been using the systems biology approaches from the very beginning itself, since biology has been started studying. So initially it used to be called as theoretical or mathematical biology. Now with the data, now with the all those omics data that we have, or other proteomics or metabolomics data that we have, this whole systems biology has taken a new phase, right? So that's how systems biology is in vogue these days in every university. So you go abroad, in every universities they have a systems biology department or systems biology research groups, right? So let's start with some basics, with what we understand for the beginners who has never heard, who has never come across this field of science, that what is systems biology. Systems biology is what we when we study the components of of any biological system, and their interactions, and how those interactions actually give rise to certain functions and behavior of that system. So those behaviors are coherent behaviors, so they keep on interacting among themselves and give rise to those behaviors of a system, right? Those behaviors are exactly, you can say, a bird's eye view of the whole system, or its function, right? Or it could be in the form of certain phenotype or physiological role of that system. So this definition is given by Neil Friedman. He's a computational scientist and also a computational biologist, who's from University of Hebrew in Jerusalem. right? And he works in the area of systems biology, and also machine learning approaches to study gene regulation. So he has coined these definitions, though there are various definitions. If you Google the term systems biology, you'll get many other definitions anyways. So the behavior of a system depends on two things, right? One is the properties of the properties of the components of the system, and the interactions between those components. So in molecular biology, what we have seen so far, the whole approach is a more of a reductionist approach, and the paradigm is shifting from that reductionist approach to the systems approach, where we have a idea of uh, uh, integrative biology, or integrative approach, or specific interactions of components in the biological systems are studied, like cells, tissues, organs, ecological webs. So there are various hierarchies at which it can be studied, starting from the molecular or atomic level, that is the cells, tissues, organs, until the ecological level. So systems biology can be studied at various levels, right? So, systems biology is a very multidisciplinary field of science. So, you all are aware of classical bioinformatics, which is always also called as a which is always been defined as a multidisciplinary field of science. Similarly, systems biology is a very advanced field, or applicative field of bioinformatics. So, that definitely it is also a multidisciplinary field of science, right? So sorry, so it is a multidisciplinary field of science. Fine. So we have we you can see in this slide that it has biology, it has engineering sciences, bioinformatics, physics, computer science, mathematics, all are integrated together. So biology definitely, because this is what is the basis here, right? We are not anyway studying any electrical system, or electronic system, or any transport system, right? So we are studying here biological systems. So though the concepts are also been the same concepts are also being used in studying other non-biological systems also, right? Or models also. But here the basic problem is problem or hypothesis or experiments are definitely biological in nature. So why engineering science is a part of, because like, if you see classical bioinformatics, we don't see engineering science concepts here, but in systems biology we see engineering science concepts here, because we use the concepts of networks, we use the concepts of control theory, we use the concepts of system sciences, even the systems based modeling and simulation concepts have come from the engineering sciences, because you must have heard that engineers design cars, design aeroplanes, design different electronic items, even these social networking sites, right? So like they are also designed on the basis of these network concepts, right? So all these concepts have come from the engineering science concepts, and now through systems biology it is being used uh, in biology, right? So, and the one of the major aspect in systems biology is networks, is studying biology using networks, right? So that is my main motto here, to tell you how networks can be used to study biological problems, or to understand biological problems. Then bioinformatics, because we use various databases, various Mr. Barua has mentioned, the different resources, like like NCBI, Uniprot, Ensemble. So all these are various bioinformatics resources, right? Basic bioinformatic resources. So all those are also being used in systems biology in some way or the other. Then we have computer science. We use various programming environments to do modeling and simulation, to create networks or network models. So we use GUI based tools, and also we use programming based tools and apps. Right, using Python packages, using R libraries, or using MATLAB toolboxes, right? So that's how computer science is a part of it as well. Then physics, definitely physics and biology are inseparable field of sciences. So dynamics, kinetics, motions. So all these are physical aspects that are important part of biology, which are touched upon in systems biology, which are being used to model in systems biology. Then mathematics, definitely when we create predictive models in systems biology, uh, we use different mathematical approaches, like using ordinary differential equations, using partial differential equations, ODE, PDs, then various probability based approaches, then then stoichiometries, matrices. Stoichiometries are anyways matrices. So matrices, kinetic equations. So all these are mathematical problems, and they are being used here to solve problems in systems biology. So I hope you understood why systems biology is basically a multidisciplinary field of science. Now, next is that in the important features of systems biology. So it has been responsible for some of the most important developments in the science of human health and environmental sustainability. So it is a holistic approach to deciphering the complexity of biological systems that starts from the understanding that the networks that form the whole of living organisms are more than the sum of their parts, right? So it is basically a holistic approach, right? So we don't study on the basis of one gene, two genes, or three genes, proteins, one protein, two proteins. No, we use hundreds of genes, hundreds of proteins, or probably the whole system. However, the uh, number of genes could be as many as 1,000, in number, or 30,000 in number. So their sizes are in terabytes anyways. So those are huge number of data in that are uh, p- uh, that are part of those systems. So studying them together is a holistic approach. So systems biology is a holistic approach. It is collaborative in nature. So I have already explained why it is collaborative in nature, integrating various basic field of sciences. And then it is its ability to design its predictive multi-scale models. So you can design it at the molecular level, atomic level, then at the tissue level, cellular level, organ level, organism level. So we talk about various organism level models, where, for example, the whole organism based metabolic models, or genome scale metabolic models of human, genome scale metabolic models of E. coli, Saccharomyces cerevisiae. So in short, we call them as GEMS. So that is at the organism level. Even uh, at the population level, the various epidemiological models, like, for example, the best example, the SIR models. SIR model is one of the example, right? So that also comes under the basic area of basic umbrella, or the overall umbrella of systems biology, right? So that's how it it is multiscalar in nature. It enables our scientists to discover new biomarkers for diseases, stratify patients based on unique genetic profiles, and target identification for various for drug designing purpose and other treatments. Systems biology ultimately creates the potential for entirely new kinds of exploration and drives constant innovation in biology based technology and computation, right? So these are some of the important features of systems biology. Fine. Now, systems biology workflow, how it actually how a particular research is taken place using such these workflows, right? So just 1 second. So you can see here the systems biology workflow. So this picture depicts the general process of systems biology. Or you can say the workflow of systems biology. It is an iterative process. So this is in cycle. So it is an iterative process which begins with collecting and integrating multi-omics data. So we start with multi-omics data here from high throughput experiments, like how you know about big data in or large high throughput data in biology or biological sciences or biomedical sciences, right? So those data can be integrated the or multi-omics or those multi-omics data can be integrated from high throughput experiments. It moves on to design and build a model. So we build a model here. This is called as model building, right? So it has some resemblance with the AI ML based approaches, but here the approaches, the various modeling formalisms or techniques are not little is absolutely different from the algorithms are different, right? So it is little different from how we study the it from a data scientist perspective. So this is an engineering perspective of or engineers perspective of studying or doing modeling or simulations in system using systems biology, right? So model building here, we do model building using various bioinformatics and mathematical approaches using computational and simulation tools. One can proceed to analyze and interpret the data. So this is model analysis and interpretation. So from analysis and interpretation, we come out with hypothesis generation, then hypothesis testing. So we do the testing of the hypothesis, and then so see here a modeling and an experimentation goes hand in hand. So it's not only solely computational in nature, but we need experimental data to validate it at every steps, right? So it starts with experimental data, but in between there are various sections where we also use experimental data to keep on validating it. So so a modeler and a systems biologist and an experimental biologist work hand in hand to come up with a model that represents the real world system accurately, right? So what is the basic aim of modeling? To make a replica or to mimic the real world system, so that we can solve and we can do various predictions, we can solve various we can understand various complex problems through those models, right? So that is the basic workflow of a systems biology experiment. So next are key properties of biological systems or models, like yes, so every biological systems, sorry, every biological systems or models have certain properties. Now these properties are all quantifiable properties. They are not just English words that you are seeing here, that irreducibility, emergence, complexity, modularity. So they all are quantifiable properties and quantifiable aspects of any biological system, so when I talk about biological systems, just for your understanding, you can think about any biological system, starting from a cell to protein complexes to cell membrane, then disease pathogenesis, to for example cell cycle, transcription, translation processes, right? Disease, like no complex diseases like neurodegenerative diseases, autoimmune disorders. So anything can be considered as a system, but every system has certain properties, right? Which I wanted to show you here in this slide, where irreducibility is the first property, where we may undoubtedly gain significant insight into each of the components of the system by studying them individually, but we need to study the system as a whole in order to gain a holistic perspective of what these components do when they are all put together in an appropriate manner. So that's what is irreducibility of a system. For example, cellular respiration, right? So you can give the example of a cellular respiration. Then emergence. Emergence is quite similar to irreducibility, but it has a different it is a different property, where systems are composed of individual elements or parts that interact in various ways. In general, the behavior of a system is quite different from merely the sum of the functions of its various parts. This reinforces the need to develop methods to study biological systems at the systems level rather than at the level of individual components, right? So next is complexity. So complexity is definitely a very important property of biological systems. So most of the biological systems are very complex in nature. So we our aim as a systems biologist to target this complexity, to understand this complexity of biological systems in certain quantities or in certain quantifiable manner. And for example, health and disease. So health and disease is one of the best example of a complex system. For example, cancer, for example, diabetes. So they are very complex disorders, right? And making a blueprint of them, that is making a model of them, is again a very havoc task, and that's what we do in systems biology. This cannot be predicted simply by analyzing the individual ligands or proteins that comprise the cells. A more complete picture of their context would be necessary to achieve it. Biological systems are, needless to say, that they are extraordinarily complex, as I said, and which is evident in individual prokaryotic cells, not only in prokaryotic cells, but also in various multicellular organisms, right? So, so for example, the insulin production, for insulin production in diabetes, or how insulin production regulates the blood glucose level in a diabetic patient. So, or probably how diabetes is related to obesity, or how diabetes is related to cardiovascular disease. So all these are complex aspects, and that defines the complexity of a biological system. Next is modularity. So what is modularity? Bacterial systems may be viewed, so modularity are modules constitute semi-autonomous entities, right? So every it's not that a particular biological process takes place in a uniform or a homogeneous system. They have heterogeneity in the system, which are called as different modules or different clusters that work together to run that system, or to for the functioning functioning of that system. So whenever I say system, that means biological system, right? So that's modularity is very important, and it can also be mathematically expressed. So all these properties can be mathematically expressed, or has been already mathematically expressed or defined, right? Then robustness and fragility. So robustness and fragility is again a very important property. So there are so much fluctuations that happen inside a cell. I'm just giving an example as cell as a system. Those perturbations or those fluctuations that comes from that are intracellular and extracellular fluctuations in a cell. So that can be in spite of those fluctuations, a system remains steady or remains or maintains its excuse me, and maintains its homeostasis. So that is called as robustness, right? And sensitivity and fragility is that with the simplest changes in the concentrations of certain metabolites, or one point mutation, or probably one small structural change in an enzyme can cause a huge difference in this whole system, right? That can cause a disease. I mean, excuse me. So that's what is robustness and fragility. And robustness and fragility is one of the another important aspect. Then there is evolvability also, evolvability of systems. So that is another property of biological systems. Now networks, let's come to our main topic, that is networks. So systems biology is huge. Right now there are many aspects of many applications of systems biology, but the one so systems biology has modeling and simulation aspects also, but modeling and simulation of the whole biological system, not just what you have studied probably in drug designing, where we do molecular modeling and drug designing. This is not molecular modeling, this is whole cell modeling, this is whole system modeling, right? We are modeling the whole system. We are not taking one module or two, one proteins or two proteins probably, and model them into so what that is a different field of bioinformatics altogether, that is drug designing, and that is called as CAD, that is computer aided drug designing, right, etc. Now here we are all trying to understand the whole system at a time. Right now, the one of the most important and one of the most preliminary study that we do in understanding the whole system is through networks, right? So networks are important aspect of studying a biological system, right? What are networks, right? What or if someone asks you what are networks, a network of complex systems can be mapped onto a graph in various ways. So networks are also called as graphs, right? So they have all come from the concept or engineering concept, that is graph theory, right? So we have so networks are basically graphs in various ways. It is possible to analyze all paths between two nodes. Now every graph or network can be defined as a set of nodes or vertices and edges. So that is nodes and edges describe a relationship between the nodes. So you can see here uh, how these nodes, or the number of paths from a node outside and the number of paths terminating at a node, are defined. So here you can see in the picture or the diagram that's below this that's at the bottom of this slide, is here we can see how nodes can be connected to each other. The figure on the right shows how A affects, how A affects C, and D also, D, but it itself affected by B and D, right? While B also affects D and C, has no connection with B or D. Again, genes, cells, again, A, B, C, D can be. What are these A, B, C, D, E? Right? No, these A, B, C, D are basically the genes, proteins, metabolites in a network, right? So I have just simplified it, or generalized it into ABC here, just for your understanding. Now, we have seen here about network, like, no, what are networks? Networks are nodes, and networks are basically representation using nodes and edges. Now we have various parameters by which those networks can be, or network topology can be parameterized or can be weighed, right? So if somebody says that how good is your network, right, you need to tell that person that your network is good on certain parameters, on the basis of certain parameters, or on the basis of certain values, right? So you give those figures, or those numbers, that shows that your network is actually quite appropriate, and it has meaning, and you can draw inferences from it, right? Network biology. So this particular topic is called as network biology. You must have heard somewhere about this subject called as network biology. So this comes under systems biology, right, where we study a system in the form of networks. So that is called as network biology. This is part of systems biology. Network biology and systems biology are not same. Systems biology is huge. There are many aspects of systems biology. We have systems immunology, we have systems microbiology, we have systems biochemistry, we have, right? So there are systems biology can be applied to any biological field of sciences, but network biology is more or less concentrated. Even in systems biology, we study models, predictive models, right? We do predictions using models, and making a model is not an easy task, right? So networks are just network represents, and it's quite interesting to get inferences from, or get insights through these networks. So network biology provides a quantifiable description of networks. There are several network measures that enable the comparison and characterization of complex networks. Now, first one is connectivity or degree. We call how well they are connected, how well a network. It's not just connecting the dots. It's every connection and every dots, how they are placed in the network has a meaning. It's not just connecting the dots. You think that network biology is just going to the string database or epi database, or probably going to cytoscape and join those dots. It's nothing. It's not as simple as that. There are various deeper in aspects of networks, where we understand networks, where we infer from networks about various understanding of those biological processes, right? Connectivity, first is connectivity. Now what is connectivity? So here K represents, connectivity is represented by K, or degree of connectivity is represented by K. So the K represents the number of links the node has to other nodes in the network. For example, for directed net, so we have two types, like directed and undirected networks. So for directed networks, K has two components, K in and K out. There are various arrows that are going out, and there are various arrows which are coming in. So everything has a meaning. So they are not like no not just webs of, or complicated pictures, but they have meaning inside them, or encrypted in them, in networks. So the in degree, so this K in is in degree, and K out is out degree, right? So they can be they can be uh, they can be quantified, or they can be parameterized by using softwares like Cytoscape. Cytoscape has various features by which you can measure these parameters, right? Then degree distribution is represented by pk, is obtained by counting the number of nodes with k like 1 2 links, and 1 comma 2 number of links 1, and dividing by the number of nodes to distinguish between the various network topologies. Then clustering coefficient is another important parameter of studying the quality of a network, where the average clustering coefficient characterizes the overall tendency of nodes to form clusters of groups, which is represented by ck. Ck is defined as the average clustering coefficients for all nodes with k links. It is also called as, we have cliques, certain cliques, or we have certain hubs. So they have different meanings in network biology. Cliques, hubs, modules, or subgraphs, so they have different meanings, right? So characteristics of path length. So when we have two nodes, there is a path between those two nodes. It measures the typical separation between two vertices in the network. Intuitively, it represents the network's overall navigability. Right now, how the network is actually navigating from one node to the other node. So the whole navigability can be represented by the path lengths. Then we have network diameters. There are huge number of parameters. We have a book, totally a book by Barabasi and Bushnan, Albert Barabasi and Bushnan, where he has explained so many type of properties of or network parameters, and every parameter gives you different meanings, different inferences, which you cannot solve at the molecular level or at the singular level. You can only solve it at the system level when you have everything in front of you, all the genes, all the proteins, all the metabolites in front of you, and you can infer from them, right? From their connectivities. So that's all about network diameter. So there is network diameter as well, where the network diameter is the greatest distance between two nodes in the network. Fine. Now, let's move ahead. So we have, on the basis of these nodes and edges and the parameters, we have various types of biological networks. We have different types of biological networks. Now, biological networks could be many types, right? Networks have no limit. Now you can make disease networks, you can make disease gene networks, you can make drug gene net drug gene protein networks, you can make so there are various type of bipartite networks or graphs that you can make. So for better understanding, you should always start with understanding the intracellular networks, right? The intracellular networks are more easier to start with. So for example, metabolic networks, we have gene regulatory networks, signal transduction or cell signaling networks, protein interaction networks, right, etc. So these are the intracellular networks. These are also intracellular networks, but they don't come under any particular category. So they are very specific, like for example, immune network or immunological networks. Depending on all the immunological aspects, you can make networks. Then there are drug target interaction networks, or DTIs. We call them as DTIs, and this particular field is called as network medicine these days. These days there is a field of science called as network medicine, or network pharmacology. So all this counts the drug target interaction networks. Fine. Right. So next is um Now, whenever we make a network in any platform, in any tool, or in any platform, like for example in Cytoscape or in Cell Designer, you make a network, right? Or right. So those networks are represented in certain formats, right? So there are file formats for networks also, or models also, systems biology models also. Networks and models are different things, right? Models are predictive in nature, networks are not predictive in nature. They cannot predict anything, they only give you inferences, and they only give you insights about the various interactions, right? So systems biology have so various formats, like for one of like no, the one of the format that one of the very widely used format is called as SBML format, that is systems biology markup language. It's an XML based format, and that is extensible markup language based format. And a representation format, it is a representation format for communicating and storing computational models of biological process. Even the networks can also be represented in SBML format. Then there are free and open standards with widespread software support. They can represent many different classes of biological phenomena, including metabolic networks, cell signaling. So all those intracellular types of networks can be represented using SBML file format. Now, next is another type of file format is there, which is quite widely used for cell-based networks, like whole cell networks. So we can make global networks of the whole cell, where the various intracellular networks are integrated among themselves, like the cell signaling, the metabolic networks, cell signaling networks, gene regulatory networks, all are integrated among themselves and make the whole setup of the cell network. So it is represented in another format called as CellML format, which is again an XML based format. And it also describes mathematical models. It is similar to SBML format, but it provides greater scope of the model modularity and reusability, and it is not specific to descriptors of biochemistry. Anyone can submit reviews, update models, and receive feedbacks and help from the community of CellML. A CellML discussion. Several other formats are also available other than CellML, like MathML is there, then BioPax format is there. So there are different other types of formats by which systems biology or network biology based models and networks can be represented and used in different softwares for various analysis and for various inferences or insights, getting inferences and insights. Now, these are systems biology databases that we use quite frequently, quite widely. First one is KEGG database. It is a collection of database with genomes, biological pathways. So it is not a pathway database. It's a very comprehensive database. This KEGG database, it has KEGG genes, KEGG proteins, KEGG drugs, right? No, so it is a very comprehensive database like NCBI. So KEGG is not. It is a myth that KEGG is considered as only a pathway database. Not, it is not correct. KEGG also has various informations or various annotations of various genes, proteins, like no single molecules also. That is single biomolecules also. Fine. So then we have biomodels. Biomodels is especially a free and open source repository for storing, exchanging, and retrieving quantitative models. So it is the only one of the sole database which contains biomodels. We have during my PhD, I created a model model that depicts the energy pool, where we I have actually integrated all the anabolic and catabolic pathways in human, right, and made a model for various predictions, you under different conditions like disease conditions, healthy conditions, fasting condition, diabetic condition, different metabolic disorder conditions. So I have tested my model and it has given exactly same results as in the experiment. So my model has been submitted and accepted by biomodels. It is available if any one of you wanted to use it. In case you are interested in that area, definitely you can use it, right? So next is big database. Big database is a BIGG database. So BIGG database is the database for biochemistry, genes, and genomes. So it contains various genome scale models, mostly the models that are being created by constraint based modeling and kinetic modeling. So those models, mostly constraint based modeling, where we generate genome scale or genomics based metabolic models. Model reconstructions or genome-based model reconstructions can be found in big database. It covers the GEMs. We call them as GEMs, that is genome scale metabolic models. Also call them as GSMMs, that is genome scale metabolic models. You get more than more than 50 models that are present for various strains or various organisms, and also human also. The whole gem of human has been is you can find in big database, right? Then we have Reactome database, which is a free online database for biological pathways. Reactome is also by okay, biomodels is a database for by EMBL EBI. Similarly, biopsych is also Reactome is also a database by EMBL EBI in collaboration with some other organizations in Europe. And Reactome database is quite opposite to biomodels. It has only the pathway informations, but it is very attractive. Reactome database, you get informations like Google Maps. Like you keep on you take a pathway, you take a network, you keep on scrolling it, and you keep on zooming it out, and you keep on entering into the intricacies of that network, into the metabolites, into the various genes that it encodes for. So it's very interesting to explore Reactome database. Then we have biopsych database, metapsych database. Then for protein interactions, we have APID database, that is agile protein interaction data analyzer. It is mostly, you must have heard about string database. So string is not the only database that you can use for protein interactions. You also can use APID database. APID is also quite a useful database as string. Probably you may not find something in string, you can search it in APID database also, right? So they both are used for protein interactions. Now we have various tools here which are quite widely used. If you are not comfortable with programming languages, you can use these GUI based tools. Like they are used quite widely, and they are very authenticated. You can publish papers by doing some good work using Cell Designer, Copacy, or Cytoscape. You are aware of Cytoscape, I think so. And you can make models in Cell Designer. You can do simulations, modeling and simulations in Copacy. And definitely you know about Cytoscape for visualization of complex networks and integrating the various gene expression or various expression data or high throughput data with those type of with any type of attribute data, right? So they are all open source databases. Other than that, you can use the various toolboxes in MATLAB. You can use various packages in Python. You can use systems biology or network biology based model making packages are there. Then you can use in R programming language also various libraries and packages. Fine. Next. we have these types of systems biology networks and models. We have four major types of networks models that can be built based on the research needs and the data in hand and the state of a system. The state of a system is a snapshot of the system at a given time that contains enough information to predict the behavior of the system for all future times. So we have these four types. Deterministic, that the future state of the process may be determined by the current state, which uses the ordinary differential equation based or PDE based methods. Stochastic, which future state cannot be determined, only a probability can be predicted. So where there are random variables, too many random variables that can be done using stochastic methods. We have continuous methods, that is called as the values of the states are continuous, where the values of the states, different states of the components are continuous. Then we have discrete, the values of the states are discrete, that is not continuous, where we use this boolean modeling or boolean modeling methods, right? Or logic based modeling methods. Now network network model development and construction. So this is the whole process. First of all, formulation of the problem, definitely. So this, you just understand this process. This is like a flow chart, or you can say these steps. Formulation of the problem, first of all you need to ask the question that you wanted to define, you wanted to work on, whether you wanted to identify biomarkers, whether you are going for drug target identification, you are going for optimizing certain metabolites in in metabolic engineering, for which are used for various bioproduct formation, optimization. So these are various problems that so you need to first identify your problem, then verification of the available information, so how much information is available for your support as a supporting data for you, then selection of the model structure, determine the general type of model, like for example deterministic or stochastic approach that you are going to use, which type of method that is suitable for your for modeling or for network based modeling. Then establishing a simple model, a model can be built through mathematical formulation. Then sensitivity analysis for validation, mathematical simulation may be highly sensitive to parameter changes. That means in sensitivity analysis we change certain parameters and see how it affects the system, and whatever is the effect in the system, okay, that needs to be validated or cross checked with the experimental data, right? So experimental tests, then we can use experimental tests of the model prediction. So we need some wet lab validation sometimes, or probably if you have a paper or literature already been done, or experiments already been done, you can compare with that and see whether your model is acting as real as possible or not, right? So then the stating the agreements and divergences between the experimental and modeling, whether the model results agree with the experimental behavior. If results do not agree, check for false assumptions, right? Oversimplifications of the model, wrong model structure, inadequate experimental data, or other inadequately represented factors. So all this will check that whether what is the issue with your model that is not giving, or it's not getting validated with the experimental data. Iterative refinement of the model, refinement of model is also very important, because the more you refine the model, the more you fill up the gaps that are present in the model, the more accurate it gets, right? Or it gives you the answer. The perturbation of a to a network model. So perturbation means the changes that we do to a model. The networks of different topologies, vary in their resilience to various types of perturbations. The process starts with systematic perturbation of biological systems, monitoring the pathway responses, integration of the data, formulation of mathematical models that describe the structure of the system and its response to perturbation. So perturbation analysis is very important, where we, for example, you knock out certain genes, you knock in certain genes, or probably you have just stopped certain pathways in a metabolic model or in a biochemical network model, and you see what changes it goes happens. If there is no change, that means those pathways are not very important for that particular problem, right? So or right, so this perturbation analysis is very important. Even you can change the concentrations, you can change the expression values of various genes and proteins and see what happens to the model, how the model behaves, right? So that is a very regular thing that we do in systems biology. Then applications of biological network models are manifold, right? So you can use it in the whole biology, any field of biological sciences, starting from the bioprocess technology or metabolic engineering, then in biomedical sciences, where we can use or biomedical applications like network-based drug discovery, discovering personalized and precision medicine, right? So these can be done using AI/ML also, but this can be done using systems biology approaches also. So the choice is yours, right? We do it in two ways. I always say that data science in biology always gives rise to two directions. One is AI/ML based approaches, and another one is systems biology based approaches. So I'm talking whole about systems biology approaches today, right? So plant and microbial metabolic engineering, personalized and precision medicine, network based drug discovery, discovering complex relations, etc. Now, network inference to understand disease pathogenesis. So this is the case study, or this is the you can say the research topic that we are going to uh, understand today. This is one of the applications that I have chosen because I have worked a lot on understanding disease pathogenesis using network inference. All right. So systems biology induced to decipher uh, decipher these multiscalar the pathogenesis of the pathogenesis. One second. This I cannot see the slide. The pathogenesis of most multi-genetic diseases involves interactions and feedback loops across multiple temporal and spatial scales from cellular to organism level. So characteristic of the network, the robust characteristic of the network um, is traded off due to impact of perturbations on the native network, leading to changes in the phenotypic responses, thereby exhibiting disease states, right? Next is clearly these network inferences, clearly the system level study is required to reveal the fundamental principles of disease initiation and progress, possible to understand the dynamics of these networks using the disease model. So you can see here the examples of disease pathogenesis networks. The first example that I can give here is cancer network. So a signaling network of remarkable complexity that is of critical importance to cancer tumor. So this is the paper that has been published. So I have taken this picture from that paper that is published in 2000 in Cell, where they have shown the complexity of the cancer cell network, right? So this is not a network diagram actually. This is just a book diagram, or you can say literature based diagram, right? So this is again a literature based diagram. This is not a network diagram. So you can make a network out of this from this keeping this diagram as a base network. Then next example is that where you can study disease pathogenesis network is diabetes type 2. It has already been published. The diabetes type 2 model. It's a systems based or network based model where you can see the various biological these various nodes are actually denotes the biological processes, protein abundances, cell secretion, the various activities actually, not the genes and proteins. The various activities has been represented with by these various nodes and edges, right? The nodes, but edges are actually here shows the either it is increasing the activity or decreasing the activity or directly increasing the activity, indirectly increasing the activity, directly decreasing the activity. So all these connectivities can be explained. So this is basically understanding the whole diabetic process or type 2 diabetes model, understanding diabetes model. So this is the work that we have done in our lab, where we have made a where we have worked on the network inferences and also drug target identifications using networks in vitiligo, or we call it as vitiligo, an autoimmune disorder. It's a chronic it's an autoimmune disorder, skin disease where the hyperactive immune system kills its own melanin cells or melanocytes, right? From the basal layer of the epidermis, which leads to patchy loss of skin color, right? Which is quite related to psoriasis, and we have also done some work where we have done the correlational networks between vitiligo and multiple sclerosis and n- then rheumatoid arthritis and psoriasis, etc. And also we are working on connecting it to other diseases like thyroid, hypo and hyperthyroidism, and then um, diabetes, etc. So we are trying to do this correlational network analysis between the because this type of analysis gives you an idea that this type of complex diseases are not caused by one factor. They are multifactorial diseases, right? So we have developed, along with that also we have developed this database, which has been published in two of the journals, vitiligo vitiligo information resource, that is VIRDB. We call it as VIRDB here. And so this is the whole structure or features of the VIRDB database, where we have differential genes, intersecting genes, natural leads of lead compounds. So we have done some docking studies also. This is just for this database we have done, though this is not our main area. So we have done some network studies, and we have shown them in the network gallery, where we have shown differential gene networks, intersection networks with multiple sclerosis, intersection networks with psoriasis, intersection network with rheumatoid arthritis, target protein networks, and also we have done some vitiligo information resource profiles like CAGDB connectivity, PDB-DB connectivity, string DB connectivity, etc. Right. So we have used mostly microarray data here for the gene network, and uh, to, we have analyzed those microarray data and done this analysis and done this network studies. We are still working a lot on different aspects of vitiligo information resource. It has more other informations. I would like all of you to visit that website. This is called as www.vitiligoinfores.com. So you can get to know our database more, and we have more such network based analysis, and also some sort of drug target identification analysis, and we have published them in PRJ and and F1000 research recently. So these are some of the features like PPI networks, GGI networks, like gene-gene interaction networks, protein-protein interaction networks, etc. Now we have come uh, almost to the end of this talk. That what is the take-home message that we can do from here? That in recent years, systems biology approaches and more specifically network-based approaches emerged as powerful tools for studying complex diseases. These approaches are often built on the knowledge of physical or functional interactions between molecules, which are usually represented as an interaction network. A variety of intermolecular interactions, including protein-protein interactions, protein-DNA interactions, and RNA interactions, are essential to these cooperative activities. These interactions can be conveniently represented as networks with nodes, with the networks that is graphs with nodes and which denote molecules or the components, and the links which denote the interactions between them. Network biology provides a powerful tool. It is a part of systems biology, but it provides a powerful tool for the study or an elegant way of studying the complex diseases or complex problems in biological sciences or biomedical sciences. Network-based approaches leverage the idea that complex diseases can be better understood from the perspective of dysregulated molecules than at the individual gene level. So I guess I'm almost at the end of my talk. Now, just one slide that shows my group, my research group at Amity University, Noida, um, in India. And we have this website. If anybody is interested, you can visit this website and see our different researches and different papers and book chapters and various aspects and various projects. We are already running a DBT funded project. We have people from here who has got DST Sub Young Scientist Awards, Wellcome Trust Genome Campus. They have been got fellowships for attending various workshops at Wellcome Trust Genome Campus in the EMBL EBI. Even I have also gone there for training, and because my model was accepted by biomodels in EMBL EBI campus, so I was selected there. So ongoing funded project, we have AI based. We also work on AI and ML based approaches also for various omics or for various big data in biology for analyzing them, right? So what we do genome scale model reconstructions, we do constraint based modeling, flux analysis, clinical data analytics and association studies, discovering biomarkers and generating machine learning models that we do separately, that is basically being headed by Dr. Priyanka Narad. She is also the another investigator in our lab, and along with me. She's also the mentor here. Multi-omics integration with genome scale metabolic models. So this is purely systems biology, multi-level biological network modeling and analysis. So these are our project associates, PhD scholars, and research associates, etc. So thank you all for listening to me, and I hopefully I could cover as much as I could in systems biology and network inferences, and how network biology can be used in solving various biological problems.
Thank you so much,
So
Dr. Sengupta. Yeah,
thank you.
Thank you very much, sir.
it was amazing to get to know so much from you. The talk was really insightful, and we explored a lot of applications about systems biology and network analysis. Well, we have a few questions in the chat from our audience, so I will take up some of them.
Yes.
This question is from Aushmita, and she asks, can the systems biology approach be used to understand the immune system of an organism? And are the similar databases used in the process like the ones used for drug discovery?
Yes, definitely. Presently, we are working on similarly as exactly for vitiligo. So, since vitiligo is a autoimmune disorder, we have not worked on the immunological networks. We can work on immunological, and this is quite new for making immunological networks rather than going into gene regulatory networks or protein networks. We can specifically focus on immunological networks for and mostly for immunological networks we use mathematical modeling or differential equation based modeling approaches from systems biology for to model them. And the databases that we use for drug discovery are different from the databases we use in systems biology. We can, yes, definitely take the if you are going for drug target interaction or drug target interaction networks, then definitely you need to use those drug databases, or probably the various databases that we use for getting the drug molecules and do some initial level docking studies to just cross check them, right? So those things can be done at that level, but definitely not for pure systems biology. We don't use drug discovery based databases that are specifically for drug discovery.
Okay, thank you so much, Dr. Sengupta. Here's one more question from Sanju Vikasini, and the question is, what are the applications of systems biology in industries? We learned about the research application, but what are the sectors in which the industries might benefit from systems biology?
Oh, okay. So, in industry, first of all, the industry that I will tell is this the antibiotic based industry, or the various secondary metabolites, or various plant based metabolites based industry, where we um, actually optimize the production of various metabolites from smaller organisms, because those organisms from where we get good metabolites or probably the secondary metabolites are very minuscule. So we use model organisms like E. coli or Saccharomyces cerevisiae to optimize their production, and that can be done, and that can be taken to the industry. For example, you must have heard about the companies like Genomatica in US, or uh, so those companies use systems biology and synthetic biology approaches for higher production of various useful bioproducts, like those are produced in the cell factory, right? So this is one of the application, and another application in health and disease is identification of drug targets, identification of biomarkers. It helps in drug discovery. Like no drug in drug discovery, we used to take one molecule, two molecules, three molecules at a time. But now using these systems biology approaches, we can work with multiple number of drug targets at a time. So that actually aggravates the importance or significance of or the more productivity of research in drug designing, right? Or drug discovery.
Yeah,
So these are
yeah,
some. There are many other. We can make models we can make various models which can be used by the various healthcare sectors or experimentalists to validate their data. So those models can also be used as um, as uh, as a model system, right? Because there are various ethical issues by for using human, for example, gem, right? As I have mentioned, genome scale model in human. So you cannot use human for human samples for every experiments. There are various ethical issues related to it. So instead of that, if you can use the human genome scale model or whole human genome scale model, like whole human genome, you can use whole human genome scale metabolic model for various studies. For example, if you wanted to study a particular type of cancer, you can take the whole genome scale model of human and study that particular aspect of cancer in using those models, those systems biology models. There are many other applications. I have mentioned few of them.
Right. Thank you so much, Dr. Sengupta. And in continuation to the answer that you already gave right now, there's another question. So John wants to know that can we apply systems biology in organ on a chip model? So that's again a substitute for testing
Yes,
anything
yes.
on a live organism.
Yes, organ on a chip model, but we use spatial based modeling. So I have never worked on that, but yes, definitely we can use. See, there is a project called as Physium Human Physium project that is an ongoing project where they generate these virtual organ models, organ based models. They generate musculoskeletal models, they generate pancreatic models, they have generated these pig heart models, then pancreas. So all those models can be generated computationally, but then they have to take it to the organ on a chip. They can translate it to the organ on a chip system.
All right. Thank you so much, Dr. Sengupta. And here's the final question from my end. So I had this question in my mind. You talked about various modeling and simulation tools. What's the computational power required to run those softwares,
Very
and how
simple.
much?
It does not require very high end systems. You can use it in your simple laptop also. The only problem is with the data. Suppose that I think you are also aware of. If you are using RNA-seq data, or probably if you are using NGS, the whole genome sequence data, then definitely you need the more computational power, like for your system for processing those data. Then we can enter into systems biology. Then does not require much computational power. But yes, for like MATLAB based, which is actually a paid software, MATLAB, and it's a heavy software. For that, probably you may need little bit better systems. But otherwise, even in a simple laptop, you can make models. But
All
yes,
right. Well,
for the initial data processing, you need uh, computational space and power.
right. I get it. Well, thank you so much, Dr. Sengupta. It was a pleasure to have you as a guest today, and we learned about so many things. I'm sure the audience got many messages to take home from today's webinar. It was insightful for all of us. Thank you so much for your time.
Thank you, Mr. Barwa.
All right. Thank you so much, everyone, for joining in. And before we wrap up today's great webinar, I have a few things to share with you, which might be interested in. Some of the more opportunities coming up, and some other ways in which you can benefit your thirst for learning. So, adding on to data science and programs on data science, we, as I mentioned in the beginning, at OmicsLogic, we also have some modules and training in collaboration with many of universities and organizations, where we talk about the data science concept and how we can apply it into bioinformatics. So I will quickly walk you through the various programs and training opportunities that we have at OmicsLogic Pine Biotech, and all of these learning training modules are held synchronously as well as asynchronously, that is live as well as pre-recorded. It is live through interactive Zoom sessions. There are various self-guided study materials already uploaded on our Learn Omics Logic platform, as well as practical hands-on assignments on the TBioInfo platform and the Omics Logic Code Playground, which I will be touching upon really shortly. This is a snippet of the way in which we have integrated the coding console onto our platform, where you don't need to download any extra IDE or a software to run codes like Python or R, which you can directly do it right over here and generate various meaningful biological results. Now, how to do that has been comprehensively mentioned in the various courses where we have courses like genomics, Python for bioinformatics, R for bioinformatics, and more. These courses introduce you to the concepts of data science in biological sciences. Biomachine learning course introduces the logic behind machine learning, what are the various types of machine learning tools and algorithms that can be used, including a tutorial on how to use Excel, Python, RStudio, as well as the tbioinfo platform. You can also go beyond these resources and write your own codes in a much more limitless way using Jupyter Notebook or Google Colab and GitHub. Through these programs, we help students understand the concepts behind unsupervised machine learning, various clustering methods, supervised machine learning, feature selection, and regression models, deep learning, and network analysis, and also combining various strategies for machine learning in general. We also have a special design of delivering training programs, which includes hands-on aspect as well as project-based learning. Learning only makes sense when you are able to apply it in a practical way, and therefore OmicsLogic trainings are different than others because it incorporates various projects examples which are taken from published research papers, which are publicly available, their public datasets, and goes through a tutorial which is designed in such a way that you will walk the steps in which a scientist or a researcher did or performed their own analysis. In this way, you will get a sense of how to define a research question, what are the various methods and techniques to apply to generate meaningful results, and finally, how to draw a biologically significant conclusion from the results that you have generated from these tools. Now, I will quickly go over the tbioinfo platform because, as we all know, biologists are not very good coders. I mean, that's kind of a usual understanding. Well, many people are expert in coding nowadays. So if you are not an expert in coding, that doesn't mean the end of the world, because at Pine Biotech and OmicsLogic, we have a comprehensive point and click AI enabled cloud based platform where you don't need to write even a single line of code. And what you can basically do is just with a click of one pointer, you can get your entire analysis done. So this is how the tbioinfo platform looks like. And just by clicking and joining the various tools which are mentioned here from the various boxes, you create a pipeline through which you can generate results from your high throughput datasets and then take it forward to analyze. The tbioinfo platform also generates publication worthy graphics and images and figures, which you can incorporate into your research paper, publication, or any posters. The tbioinfo serves various purposes, which includes the sequencing RNA-seq domain about various machine learning tools such as unsupervised, supervised, and more, and variant calling, structure analysis, phylogenetics, and a lot more than that. So here's again talking about how these OmicsLogic courses not only teaches you to understand the logic behind bioinformatics, but also teaches you to get expertised with Python and R. So these are some of the courses that we already have in our on our platform, which you can complete at your own pace whenever you want. So Python, R, and finally you can translate your understanding and your knowledge to basic and translational research projects that you can do. So the entire story winds up like this at OmicsLogic. You get training, you learn some concepts, you learn some skills, then you apply it in your own project, and you can be someone who can benefit from our ongoing research fellowship program, which is equivalent to the internship programs that you have all across the world. By completing the fellowship program, you will be able to design your own research project with the help of mentor guidance at Pine Biotech. So here's an example of a project completed by a student from Amity University, Noida. Her name is Vedika Rai, and she did her project in the transcriptomics of COVID-19, which is now published on our platform. As Dr. Sengupta also mentioned, we have a huge collaboration between Pine Biotech and Amity University, And as a result of this collaboration, especially with the Amity University Noida campus, we bring various exciting opportunities for the students of Amity University, such as industry academic internships and more. So apart from the already uploaded asynchronous courses that we have on the on the OmicsLogic Learn platform, we also have various programs which are launched time to time. In these programs, you will get training from our mentors who will give you their insights into some specialistic aspect of various applications of bioinformatics. For example, right now we are running a bioinformatics for infectious diseases program where you will learn about how you can apply bioinformatics for research in the field of infectious diseases, specifically taking the examples of COVID-19, Ebola, malaria, and tuberculosis. In the future, we will also be having specialized courses on precision oncology, where we will be talking about cancer research in more depth. For anybody who is looking to start with a journey in bioinformatics, you are most welcome to check out our Introduction to Bioinformatics program, which introduces you to the various concepts of bioinformatics. We also have programs dedicated to genomics and genomics in the virtual lab. We also will be having specialized programs on transcriptomics and data science for biomedical data analysis, such as the one that in which we will be talking about all the concepts that we were discussing today. We also have a major collaboration with the Federation of Asian Biotechnology Association, or FABA, and as a result of this collaboration, we will be coming up with various exciting opportunities for the students in India and Asia as a result of the huge network of industries that FABA is associated with. Talking about our international collaborations, we also have a massive collaboration with the Louisiana Biomedical Research Network and Louisiana State University, where we are providing courses and training programs for the students who are a part of this institution. Talking about the National Association of Idea Principal Investigators, also known as NEPI, we are providing free bioinformatics education to 23 states in the United States of America and Puerto Rico. And as a result of this major collaboration, many students in the United States of America are benefiting in the domain of bioinformatics education, research, and training. So it's a good opportunity for requesting all of you to stay in touch with us. We are available on all the social media handles. Please find us and stay in touch with us to get notified about any upcoming opportunity where you can take part and benefit from. A quick reminder about Sci-Omics. So we were having various topics throughout the many months, and in February we had bioinformatics and data science. We will be having many more sessions coming up in the next weeks, and in March, specifically on 31st March and 1st April, we will be having a huge international conference which is known as the Omics Research Symposium. This is a great opportunity for all of you to take part in. In this symposium, we will be having special guest speakers from all around the world, from various academic partners, from various universities, from various industries, and also research labs, who will be sharing their research and progresses made all throughout the past year and the applications in bioinformatics, leveraging it for performing cutting edge research. We will be also having various exciting opportunities for students, namely the poster presentation competition, where you will be given the opportunity to submit your own research as a poster. These posters will be evaluated by our judges, and you will be getting prizes at the end of the poster presentation competition. More information about the symposium, the poster conference, and the panel of speakers and the dates and times will be available on our social media handles as well as on your email. So keep an eye on all of those channels and platforms. Finally, it's a request to all of you present here to kindly scan or copy the link and give us a feedback of how your experience was in this particular webinar. Your feedback is important for us because it helps us provide you quality learning experience in a very smooth and seamless way. So here's a heads up for all of you. In coming up, then in the next Sci-Omics session, we will be having Dr. Arska Soek as our guest speaker. She is an assistant director at the Louisiana State University, Shreveport, and she will be talking about bioinformatics and data science training and how She and her group is revolutionizing the way in which bioinformatics is provided to the students and how students from any background can learn and apply bioinformatics in their research. So this webinar will be held on February 11th, 2022 at 3 p.m. Central Standard Time. And on February 18th, we will be having Dr. Mohit Mazumdar, who is a business development director and project mentor at Pine Biotech. And he will be talking about structural biology, its research aspect, as well as how you should design your research project and your abstract. This is in lieu with the poster presentation competition. So I encourage all of you to take part in it and attend these sessions so that you can get the insights from our experts about what's the best way to design your abstract, what's the best way to design your project, and that would help you be in a better position to be awarded the winner of this poster presentation competition. Having said that, we are now at the end of today's webinar session. I will take up all the questions that you have in the chat. Feel free to use the chat section to post more questions. I will be definitely answering all of you. So about the recording of today's session, you will be definitely getting the recording through your emails. Please keep an eye on that. And also on our social media handles, we will be posting the links to the recordings. Talking about the training in Africa and Nigeria, we do have various training programs that are running in Africa. We have our team of OmicsLogic at Africa who are based in Africa and who are providing quality training and education. So if you're interested in learning more about those trainings and how to connect to the group, please send an email to us at marketing@pine.bio. I will put it in the chat, or you can also send it to communications@pine.bio, and we will provide you with further information regarding the training in Nigeria. I will share the link of the feedback form with you so that you can go over with it. Just give me a moment. Okay, so the WhatsApp group was full. I will check it and I will send you further links regarding the WhatsApp links, and I will tell you about the poster presentation session in just a minute. Just give me one moment. I will share my screen and I will show you what the poster presentation session looks like. So while I am doing that, the poster presentation session basically is for students who are doing your own research or you want to do a research. You are welcome to submit your abstracts for now. And once you share our submit your abstracts, the deadline is 28th of February. Once you submit your abstract with us, we will be doing a screening of those abstracts to identify the best ones. And once we have identified them, we will definitely contact you regarding the guidelines for sharing your poster. And I am resharing my screen to enable you the poster presentations link. So this is the page for poster presentation. I'm also putting it down in the chat so that you can also visit it. So the poster presentation for the OmicsLogic Research Symposium covers these following topics: omics and bioinformatics, obviously applied in research. You might have done it as an independent project or a project with your university professors or your summer internship somewhere. You're welcome to submit such projects. If it is in the domain of AI and machine learning and data science, that is the application of data science, AI, and ML into bioinformatics and biological science. We are also interested in welcoming abstracts in the domain of astrobiology and spaceomics. That is a new and exciting domain because we will be having a separate panel in the symposium dedicated to astrobiology and spaceomics, and various great speakers from the most famous organizations in space sciences and astrobiology will be coming in this symposium. So it's a great opportunity to give exposure to your research and interest in front of those experts. And finally, if your research is in the domain of computational biology and biotech and pharma, you're also welcome to submit your abstracts here. Now here's a quick guideline for the abstracts. So the abstract length can be from 150 to 200 words. You should divide it into various segments such as objective, methods, results, and conclusion. The font should be in Montserrat and 12 points, and it must be a PDF file. So you have to convert it into a PDF file and submit it using this particular form right here. Just go down, you will get an option to choose your file and submit your abstract here. The deadline is 28th February, so keep that in mind. Here are the three prizes that you can win: the best poster presentation, the best citizen science project, and Young Sheldon Award for high school students who are doing cutting edge research in the domain of bioinformatics. So that's all that I have to share about the poster presentation. If you guys have any further question, please feel free to put that down in the chat section and I'm happy to take it up. If not, then that brings us to the end of today's Sci-Omics session. I had a great experience talking and interacting with all of you. We definitely learned a lot and got some great insights into systems biology and network analysis from Dr. Sengupta. Thanks to Dr. Sengupta once again for his time. And if you want to contact us anytime later on, please feel free to send me an email in the email IDs that I have shared in the chat, marketing and communications @mind.bio, and I will be happy to answer all your questions. Having said that, that brings us to the end of today's Sci-Omics session. We will again meet on February 11th at 3 p.m. CST. Until then, take care. Thank you so much for joining us, and I wish you a very good evening. See you soon. Yeah, Vadhi, I will send the link to WhatsApp by email when I share the recording of the session, so you can use that to join us.